we're going to be talking about nucleic acids, which is going to be our last form of biomolecules. And this is going to be fairly straightforward. It's probably going to be stuff that you remember from freshman year. But we're just going to kind of do a general overview. And the reason for that is because later we talk about DNA and protein synthesis. And so we're going to go into a lot more depth whenever we hit that topic. And so I did forget my iPad, so I have kind of this little setup here. So hopefully you can see what it is that I'm writing. And I will also post a copy of my completed notes. That way, if you can't see them, you'll be able to look at those to be able to see, what it was. But definitely watch the video so that way you can hear me explain the important aspects of nucleic acids. All right, so let's get started. So when we're talking about nucleic acids, they all have monomers that are called nucleotides. And so nucleotides are made of 3 units. Okay, and those units are a phosphate group, a sugar, and a nitrogenous base. Okay. So one of the things that's going to be the same for all of these is that, they're all going to have a phosphate group and they're all going to have a sugar. It's the nitrogenous base that's going to vary depending on whether you have DNA or RNA, and also depending on what protein or what gene you want to code for. And so let's go ahead and talk about, what a nucleotide actually looks like. So right here, I have this little picture of a nucleotide, and I can see I have a ribose. Okay, so if it ends in ose, it's a sugar. I have my phosphate group that's right here, and then I have a nitrogenous base called cytosine. Okay, so these are 3 different parts. So remember, my cytosine, that's the part that can vary. Okay, so I can do adenine, I can do thymine, I can do guanine, I can do uracil, and so cytosine is the one that is going to vary. But I will always have a phosphate group and I will always have a sugar. So based off of this, I think I got ahead of myself, but that's okay. Okay, based off of this, I can tell if it's DNA or an RNA based on the sugar. Okay, so DNA is going to have deoxyribose, while RNA will have ribose. And so because this one has ribose as the sugar, I know that this one's RNA. Next, it asks, is the base a purine or a pyrimidine? So I'm going to hold off on that for a second because I do need to talk about this part over here where it actually discusses purines and pyrimidines. I knew I got ahead of myself. So I'll go ahead and go on up here to the corner. So I have pentose monosaccharides. Okay, so pent, okay, that's a prefix for 5. Ose means sugar. Okay, mono means 1. And saccharide is also sugar. Okay, so I know that this is going to be a 5-carbon simple sugar. Okay, so 1 ring, but with 5 carbons on it. All right, so if I look at this pentose monosaccharides, I have 2 types. I have ribose. Okay, so ribose is found in RNA. And then deoxyribose is found in DNA. Okay, and you can see they look very similar. All right, so if I'm actually looking at this, the only thing that's differing is this right here, okay, that hydroxyl group that's in this corner carbon. Okay, so deoxy is without oxygen. Okay, so it's not going to have that hydroxyl group. You don't need to know that, but just in case you were wondering. All right, so ribose found in RNA, deoxyribose found in DNA. So I'm going to go ahead and scoot on down. So I also have different nitrogenous bases. So if I'm looking at these nitrogenous bases, I can see that they're very similar in structure. They're all rings, but I have 2 that are double rings, and then 3 that are single rings. Okay, so 2 double, 3 single. So the ones that are double rings, guanine and adenine, these are called purines. Okay, so 2 rings is a purine. Okay, that means that these guys down here, so cytosine, thymine, and uracil, these are all pyrimidines. These are all 1-ring structures. Those are all pyrimidines. All right, so purines have 2 rings, pyrimidines have 1 ring. All right, so when we're talking about our nucleotides or our nitrogenous bases, DNA has adenine and thymine, okay, an adenine and thymine match, and guanine and cytosine, okay, and those match. So you're not going to see adenine pair up with cytosine or guanine, you know, and thymine is not going to pair up with guanine or cytosine. And the reason for that is if we look at these bonds that can be formed, the bonds have to be able to match. All right, so we'll talk more about those bonds whenever we hit DNA and RNA, but the reason for that is just because the shape, you know, would throw it all off. So if DNA has adenine and thymine and guanine and cytosine, RNA, okay, also has adenine, okay, has guanine, cytosine, but it doesn't have thymine. It has something called uracil. Okay, so we're going to abbreviate that as U. All right, so RNA has adenine, guanine, cytosine, and uracil. So now that we've been able to look at this, okay, 2 rings, purines, 1 rings of pyrimidine. I go back up here to cytosine. You can see cytosine is 1 ring. So I know cytosine itself is going to be a pyrimidine. Right? So one of the things that we're also going to talk about later when we talk about our nucleic acids, so DNA and RNA, we have different types of phosphate groups. Usually you just have a monophosphate in that background, but it's kind of odd. So DNA and RNA, when it needs to form, it actually needs energy in order to form. And so essentially what's going to happen is that DNA and RNA are going to have extra phosphate groups. Remember I told you, breaking a bond gives you energy. So that DNA and RNA that's trying to form has the extra phosphate group. It's going to snap off a piece of that phosphate and it gives it energy to form the DNA and RNA. So it's kind of cool. It's almost like our DNA consists of all these tiny little bombs, especially when it's, you know, trying to be made. And so that's something that's really neat. So next, I'm going to go ahead and talk about DNA. Okay, so like I said, hopefully that was a good review. Okay, so DNA, we think about the function of DNA. Okay, we know it holds genetic material. Okay, holds genetic information. It carries is probably a better word, but that's okay. Okay, so essentially these are all the building blocks for the cell. How the cell is going to look, how it's going to function, what structures it needs, how it's going to perform, things like that. Okay, that's what DNA does. And so the DNA in our cells is kept in the nucleus. Okay, so found in the nucleus of eukaryotic cells. All right. So, and you could tell yesterday in between my classes, I went ahead and colored. All right. So when we're talking about our base pairs, remember I told you about the bonds, okay? The bonds have to match up. That's very important. So I could see right here, I have adenine on one strand and thymine on the other. I'm going to have hydrogen bonds that's going to link these guys together. Okay, so between adenine and thymine, I can only form 2 hydrogen bonds. Between guanine and cytosine, I'm going to have 3 hydrogen bonds. Okay, so it's like a puzzle piece. Adenine has to go with thymine, guanine has to go with cytosine. If not, you're going to have an extra, you know, bond that's empty, just kind of hanging out right there. And it's not going to bond to anything. So that's why they have to match. All right. So when we're talking about these particular complementary base pairs, okay, so when we're talking about complementary, we're talking about matching. Okay, so that's what this means. Complementary means matching. Okay, so it's not a compliment like, oh, your shoes look nice today. Okay, it's complementary like it's matching. And then not only that, if you look at these letters, okay, so these letters are all right side up. On this side, they're upside down. So the bases are actually going to run anti-parallel to each other. Okay, so what that means, okay, so one strand is upside down. So runs anti-parallel. Okay, oh, can't even see it. There we go. All right, so next, okay, so down here, you have a little key if you want to print this out and you can color it yourself. For the structure of my DNA, you could see it's double-stranded, looks kind of like a twisted ladder. The outside has this phosphate sugar backbone. Okay, so phosphate sugar backbone. The inside is where my nitrogenous bases are located. Okay, so I have a twisted double helix. Okay. The backbone is alternating phosphates and sugars. And remember, the sugar for DNA is deoxyribose. The rungs of the ladder, okay, the part in the middle, are nitrogenous bases. Okay, so for DNA, we have adenine, thymine, guanine, and cytosine. Okay, we don't have uracil for this one. All right, and then, okay, in between the backbone, okay, our important bonds are called phosphodiester bonds. Okay, so phosphodiester bonds hold the backbone. Hydrogen bonds connect the bases. Okay, so that's probably the newest part, is the fact that we have something called a phosphodiester bond. Okay, so I just think, oh, there's a phosphate group, phosphodiester bond. Okay, because I know that's the one weird bond that we have. All right, so last thing we're going to talk about. Okay, so one of the things that is different from AP Bio versus freshman bio, okay, is that RNA actually has a huge role. Okay, we're going to talk about it all the time. Okay, RNA, like, it is so big that scientists actually believe this is how life evolved. That's how big RNA is. Okay, so we're going to talk a little bit about that. And so we do know some things about RNA. So for example, we know it's shorter, and so its main purpose is usually to carry genetic information. So like, for example, it's going to carry it from the DNA wherever it needs to go. Okay, so RNA, usually shorter, to carry, let's see here, genetic info to other cell parts. Okay, so, okay, that's something that's special. We also have a special type of RNA called ribozymes, okay, and they essentially act as enzymes. So some RNA act as enzymes, and these are called ribozymes. Okay. So in case you don't remember what an enzyme is, it's something that speeds up a chemical reaction. Okay, so typically enzymes are proteins, and so we just say that they catalyze a reaction. They make it just go faster. Okay, so we've discovered that there are certain types of RNA that function like that. They're going to speed up that reaction. And the reason why I'm mentioning this and why ribozymes are important is because there are some ribozymes that can actually self-replicate. Okay. And additionally, some of them can stick to amino acids, and so that helps catalyze any type of amino acid reaction. So that's something that's really important with RNA. So something that, you just need to, keep in mind. And so I'm going to talk about the RNA world hypothesis. We're actually going to talk about this again when we hit evolution, but it's very important. Because some of these RNAs can self-replicate and they can catalyze reactions, scientists actually believe that RNA was the very first genetic information molecule in very primitive organisms. There's even hypotheses like that, ribozymes actually didn't even exist in organisms. It was like in this primordial soup, and they were taking all these chemicals and building, and breaking down. So they were building polymers, breaking down polymers into monomers, and they were just doing it over and over again. And so we've actually been able to replicate this. So it's kind of unusual. And so scientists believe that these guys were the very first thing that resembled any type of life on Earth. So because ribozymes self-replicate, scientists believe primitive RNA was first, genetic molecule. Okay, so they can self-replicate, and they can make and break the macromolecules. Okay, so that was something that was really interesting. All right, so let's go ahead. The last thing we're going to look at is our structure. Okay, so the structure is very similar to DNA. Okay, so we can see these in single and double-stranded forms. I feel like my handwriting is particularly bad today. I apologize, guys. All right, so also, okay, we have a sugar phosphate backbone, except for in this case, instead of deoxyribose, we have ribose. Okay, so sugar, phosphate, backbone, and it has the bases sticking out. Okay, they're not bonded to anything usually. Okay. So in the event that they're bonding to something, it's typically because they're transferring a message, they're relaying a message. And so they can temporarily bind with other RNAs or other DNAs. Complementary DNA or RNA. Okay. All right, and again, if you want to color, there's a little coloring, what's it called? A little coloring guide right there. Okay, so hopefully this helps. Now you understand a little bit, why RNA is so important, and we're going to be talking about it again and again and again. If you have any questions about nucleic acids, refer to your textbook, ask a friend, or ask me. All right, so hope you all have a great weekend, and I will see you on Monday. Bye.